Ugh. All right, let's see here. All right, we have a small group today. I'm sure people will trickle in, but they got the reprieve of not having to come in yesterday, and they were probably like, I'm gonna stay home. Good for y'all. All right, so today we're gonna go through and finish structure and function with a little bit of genetics discussion for the first kind of couple hours, and then we'll move into disease and dysfunction, where we will stay and live for all of next week. I just wanted to redirect people to the Moodle page, just because I've had some questions about where things are kept. So each week has a schedule, and if I try to update topics as I can. And then under here, this is where I'm posting the videos. They're in these little like booklets. So they're all located there from the days, and you can just scroll through and rewatch. And then all of the resources are located in folders. So any resource I'm sharing or referring to. So if you're having trouble finding things, I direct you to this page. I think that would be the best place to go. I will do my best to update next week's stuff over the weekend as well. And when I say over the weekend, I mean today. So if it doesn't happen today, it probably won't happen until Tuesday. So I'll do my best to remember to update this and put resources here. All right, we're going to get into some genetics. My first little lecture is just a basic like intro to genetics lecture. I wanted to just have a few baselines for us, mainly so we can talk about recessive and dominant autosomal inheritance patterns and sex-linked or X-linked recessive inheritance patterns. There's not a lot of basic genetics questions on the NPLEX, but if there are, they tend to be like Punnett square based. Can you do a basic Punnett square and figure out inheritance and some simple probabilities? So we'll look at that today, and we'll do like basic math. When I say basic math, like 50%, 25%, things like that. That's the maximum they're going to do. They're not going to give you a pedigree. They're not going to make you do a dual linked gene. They're not going to, make you do any of those crazy interpretations, but they could ask, what's the likelihood of offspring to have whatever autosomal recessive disease? And then they'll give you if the parents have it or carriers. So some definitions to get us going. Again, reminder about our diploid number. So diploid number is the 46th number of chromosomes in all cells except for our gametes. Gametes have just 1/2, so they have 23. The 46th number we get to because it's 23 pairs of homologous chromosomes and one pair. So it's one pair of sex chromosomes, either XX for female or XY for male in a non-pathologic sense, and 22 pairs of autosomes. That guide expression of the majority of traits. The karyotype is our chromosomal complement displayed in homologous pairs, and the genome is the whole meal deal. Our alleles, when we start hearing other words, you could potentially see homozygous and heterozygous on your test. So homozygous are your alleles that are the same for a single trait. The DNA sequence is the same on both homologous chromosomes. Or heterozygous, the alleles are different for a single trait. So we'll often see that referred to as big X, that would be the same, little x, that would be the same, and then big X, little x, that would be different. So homozygous, heterozygous. And we can see this across the board. My little eraser. All right. Dominance. So when we talk about dominant versus recessive, dominance is just essentially one allele is masking or suppressing the expression of its recessive partner. Dominant alleles are denoted by capital letters versus recessive are same letter but lowercase. So you often see an uppercase J, lowercase j, uppercase B, lowercase b, et cetera, et cetera. The dominant trait is expressed even if the other allele codes for a recessive trait. So if you have a dominant trait, that's going to pass through. That's why autosomal dominant diseases are always going to be expressed in someone who is a carrier, because the dominant trait, you just need one to express said trait. Versus a recessive trait is expressed only if both alleles are recessive. So for our autosomal recessive disorders, you need both of your chromosomal pairs need to express that recessive trait to express the disease. If you have a recessive trait and you have the dominant trait and the disease is a recessive disease, you're a carrier, but you wouldn't actually express the allele or express the disease, but you could pass it on to offspring. Your genotype is your genetic makeup of the person for the trait. Your phenotype is the physical expression of the genotype. So the genotype is how we're talking about documenting things, uppercase J, uppercase J. It's the genetic information that describes what's happening. The phenotype is what that looks like physically, whether it's a characteristic, whether it's a condition. That's your phenotypic expression is what we could typically see clinically, versus your genotypic expression would be what we would look at if we looked at your genome. Okay. Every person in the world has a unique set of genetic variants, and this is due to several different things: independent assortment, crossover, and random fertilization. And we're going to look at independent assortment and crossover. I've already defined crossover a bit, but we're just going to relook at it from a genetic lens, not from a lens of oogenesis or spermatogenesis, which is what we looked at it last time. So there's two important separations of traits that occur during meiosis I of gametogenesis. Segregation, essentially two alleles of one trait will be separated and distributed to two different daughter cells. So if you have a dominant and recessive trait, you are the carrier of a recessive trait. The dominant will go to one daughter cell, that chromosome that carries that dominant trait, and the recessive trait, that chromosome that carries the recessive trait, will go to another daughter cell. If you have issues with segregation, you can have cancer, infertility, Down syndrome, etc. We think about trisomy as there's multiple trisomies, which we'll look at here today. There's 3 that you should know, not only Down syndrome, but if you have essentially inappropriate chromosome separation, you can see potentially some of these conditions like a trisomy, or you can even see conditions where you may have lack of compatibility with life if you actually don't have appropriate separation of those chromosomes or chromatids. Okay. Independent assortment of 2 different traits on 2 different chromosomes. This one we won't talk as much about. I said they're not going to really talk about traits that are inherited together, luckily for you. So there's not going to need to be any 8 trait crossover exciting Punnett squares. So really only talk about 1 trait singularly at a time. But in reality, there's multiple traits that are kind of inherited together as like a package. And so it's not just 1 trait, 1 chromosome, 1 place. In reality, it's like multiple traits on an area of the genome or an area of the gene that encodes for a multitude of different disorders or conditions or natural states, and they will actually move together. And so if there is a disruption of that normal movement, that could lead to pathology or abnormalities. Okay. Just a reminder of what things look like here. So we have our, this is in meiosis I, we're in metaphase here, so metaphase I, and then we have separation from one side to another, and you can see that there's some variation, genetic variation, depending on the maternal or the paternal chromosomes. So crossover is what we've also talked about. So during crossover or chiasma, chiasma is also, or chiasmata is the point of crossover from these chromosomes. That's where we get the sharing of genetic information. And this is where you essentially will have 2 separate chromosomes that will share information and create a new chromosome that looks different from the 2 previous before. If you have breakage or damage or some type of dysfunction in this process or in this chromosome, you can see a recombinant chromosome or a recombinant issue that is either helpful or not helpful. So breakage that leads to normal expression of genes, helpful, fine, genetic diversity, good for natural selection. Breakage or recombination that leads to a mutation or a gene editing error, or maybe breaks in the middle of a gene sequence or genome sequence, that could lead to pathology or disease. And so that's kind of how we can get some of these mutation processes. We can also see, and we'll look at a few diseases later today, where you'll see these genetic code repeats that can occur. And that is an error, that's an inborn error in the genome and genetic code and replication. And so it depends on all these processes for how this actual inheritance pattern and congenital diseases will take form. And this is just showing if you're visual, showing how these 2 are essentially passed typically on the same genome, but we're doing some crossover. So we're going to have some exchange there. And so we have a new segment of gene or DNA. Beautiful. There's very few phenotypes that can be traced to a single gene. Typically, there's phenotypes are expressed on multiple different genes and sometimes on multiple different chromosomes to protect from inborn errors of metabolism or genetic variants. When we are talking about inheritance patterns, we will use a Punnett square. And so for those of you who've forgotten or who have tried to forget a Punnett square, you literally just make a little box. And then whatever we have, our genotype is going to be here and here for our maternal and paternal chromosome, or maternal and paternal genes. And it doesn't matter which one you put on which side. You just put one on one and one on the other. We'll do some examples here. So if we're going to use albinism as an example, our dominant allele is going to be capital A. That would be normal pigmentation. Our recessive allele, which encodes for albinism, is going to be a lowercase a. So if you had both of the same, those would be homozygous. If you had one of each, that'd be heterozygous. So let's say we had a parent who was homozygous dominant, and we had a parent who was homozygous recessive. This means that which parent would be expressing the trait for albinism? Yep, the recessive, absolutely, because it's a recessive, autosomal recessive condition. So when we actually, if they decided they wanted to have a child, you're going to take one from each. Essentially what I'm doing is I'm pulling down this capital A and this capital A, and then I'm pulling across this lowercase a and this lowercase a. And you'll see that 100% of the offspring from this cross will be carriers, but 0% of the offspring will actually express the disease because it's a recessive autosomal condition. So this would be a 0% expression or no chance of passing this to offspring if you started with this genotype. Now, if we look at a different scenario, sorry, May, I should have brought my own markers today. A different scenario where you have a recessive parent, and let's say you have a parent who doesn't express albinism, but who does some genetic testing because they want to be sure they don't pass on this trait to their children, and the parent who doesn't express albinism is actually a carrier. That's when you get more concerned. So then you would want to look and see, okay, so we'll bring our big A's down, our little A's down, and our little A's across, and we end up with a 50% chance that their offspring will express the trait for albinism, or will essentially have that condition. Because 50% of them are going to be homozygous recessive, and 50% will just be carriers. The last one you could see that often comes up with our autosomal recessive traits are if they're both carriers. Both parents, neither of them express the trait. So this would be a question like, neither parents express the trait, but a child, they had a child or their offspring expresses the trait for albinism. What is the probability that that would happen? Okay, and so then you know, okay, both parents did not have the condition. They didn't have albinism. I know albinism is a autosomal recessive. So the only way that both the parents could not actually have the condition, but have an offspring that does, is if they're both carriers. And so then what's our percentage? Well, we can bring our big A down, bring our little a down, bring our big A across, and bring our little a across. And you can see that 25% or 1 in 4 offspring would have the trait or carry the trait for albinism. 75% would not. Of those 75%, 2/3 are going to be carriers, okay, or 50% total. So these would be your most common Punnett squares that you could see utilized in an autosomal recessive case. So really your logic is thinking about, is this autosomal dominant versus recessive? We'll talk about sex linked here in a moment. And then from there, what were your parents? What were the parents, or who were they talking about initially? Did they have the condition? Did they express the condition? Did they state they were a carrier? Did they talk about offspring? Did the offspring have the condition, express the condition, state they were a carrier? And what are they asking for? Are they asking for percentages and percentages of carriers, percentages of people who don't have the condition or trait, or percentages of people who do? They will ask any form of that on NPLEX. So those questions, it's important that you read for what they're asking for, because they'll often include 25, 50, and 75%. So you really need to know what you're looking for, or they'll do a 1 in 4, 2 in 4, whatever. Questions on these kind of basic Punnett square things, reminding you of Gen Bio? back in the day? Yeah, okay, right. And this is just an example, kind of looking at this here. And then kind of, again, talking about ratios, we're not going to flip a coin. They are not going to make you do a lot of excessive probabilities. So I would say one Punnett square is about the maximum they're going to go. They're not going to have you do multiple probabilities and make you add or multiply or remember those math. There's no chi-squares on this exam. So none of that I would worry about. I'd just be able to do the simple math of 1 and 4, 2 and 4, 3 and 4. So dominant traits, so if this was switched for a dominant trait, right? We would be looking at if we had someone who expressed the trait, was a carrier, and someone who did not, we'd have 50% of the offspring would express the trait and 50% wouldn't. In this instance, 100% would express the trait because it's a dominant, autosomal dominant inheritance pattern. And then this one, 75% would. So again, knowing if the condition is autosomal dominant versus recessive is really key. And that's what I would focus on learning more than even the genetic like SNPs or specific numbers and letters and all those pieces. I would know the common conditions that are autosomal dominant and autosomal recessive. And if you remember those inheritance patterns, the majority of actual genetics questions you come across, you'll be able to work your way through. There will be some that will be on the numbers and letters. We'll look at those common ones. Recessive inheritance patterns, again, we've talked about those. We will move into sex-linked here in a moment. This is looking at some genes that are dominant or recessive. Cystic fibrosis, albinism, Tay-Sachs, the absence of Huntington's disease. That's kind of hilarious because Huntington's disease is a dominant gene, so don't confuse yourself with that one. So funny. But our dominance, we have Huntington's being probably the biggest one we discussed. So I would say, and I have an acronym or a mnemonic that I'll show you at the end of this lecture. There are things like incomplete dominance that exist. Trying to think. Sickle cell trait is kind of when you see this the most. So sickle cell anemia or sickle cell trait is when you could see this come up on your test. Essentially, incomplete dominance is those heterozygous individuals, those that have a capital and a lower letter, those that have a dominant and a recessive trait on one of their chromosomes each, will have an intermediate phenotype. So it's not that they have the condition, it's not like they don't have the condition, they have a combination, right? So there's, instead of 2 phenotypic expressions, there's 3. These individuals might have some symptoms, but they're typically less extreme than those who would have the homozygous presentation. So for the instance in sickle cell or the sickling gene, normal hemoglobin is made with 2 dominant traits. So right here, if you carry the sickle cell trait, you'll see both mutated and normal hemoglobin is made. And the person can have a sickle cell crisis if you had prolonged reduction of blood oxygen. But it is typically less common and it's less severe. These are patients who may be living with sickle cell trait for a long period of time, and until they put themselves in a hypoxic state, never knew that they had the condition or carried the trait or had the disease. Sickle cell anemia is when you carry both the homozygous recessive traits, and then you're only making mutated hemoglobin, and the person is going to be much more susceptible to sickle crisis, even in a short reduction of oxygen, because all their cells are creating essentially this mutated hemoglobin form. Does that one make sense? Or do you have questions about incomplete dominance? You still do the Punnett square the same. You're just now looking at, instead of 2 phenotypic expressions, right? Don't have the trait or have the trait. For like autosomal recessive, you have 3. You have don't have the trait, kind of have it a little bit, and have it fully, if that makes sense. Or if it was a dominant expression, it could look different as well. Typically, you see incomplete dominance with recessive traits. So if you had homozygous recessive, that'd be full expression. And then if you're heterozygous, that'd be partial expression. Sickle cell is the one that I've seen them test on the most in regards to this presentation. You can also have multiple allele inheritance. That's your blood groups. So your ABO blood typing, because you can express antigens for A, antigens for B, antigens for both A and B, or you can have the absence of all. That's how you get type O blood. So just understand that this is an example of multiple allele inheritance because you'll have a different phenotypic expression and a different genotypic expression depending on the actual alleles or the antigens you have been exposed to or that your blood type carries. And then this is just talking about what I already said here. So OA is type A, OB is type B, AB is type AB, and II or OO is O blood, O type blood, the lack of expression of both of these types. Have I ever seen them make you do a Punnett score for blood typing? No, I've more seen them ask these questions in regards to like fetal development, but typically I see these on NPLEX II because they're talking about ROGAM or therapeutics or things of that nature. We'll talk a little bit about it when we talk about immunity next week and hypersensitivity responses, because that's when this kind of like works into this intolerance. Now, sex-linked inheritance. These are inherited traits that are determined by genes on your sex chromosomes, either your XX or your XY chromosomes. So all of these other things we've been talking about were your 23 autosomal chromosomes, or 22 autosomal chromosomes. That's all of these autosomal inheritance patterns. You only have one pair of sex chromosomes mixed in your 43 total chromosomes or your 23 pairs. So this is just referring to that one sex-linked chromosome pair, your XX for females or XY for males. So your X chromosome has tons and tons of genes on it, encodes for lots of proteins that you need for brain function and for quality of life. Your Y chromosome carries much less. And you'll actually see here, the X chromosome looks much more like what we expect a chromosome to look like, and the Y chromosome is much smaller in its appearance. And so that's why it encodes for less genes. It's just physically smaller chromosome. Because of that, fewer regions can participate in crossover. Genes found only on the X chromosome are called X-linked genes. And typically, not all the time, but typically when we're talking about sex-linked inheritance, we're talking about an X-linked disease on the X chromosome. It's not always the case, but that's pretty much all that I see tested on your NPLEX exam are X-linked and typically recessive conditions. So X-linked recessive alleles are always expressed in males and are never masked or dampened because there's no Y counterpart. Again, in a male, they're only going to get, in a male that has normal XY genome, right? They're only going to get one X chromosome. So if that X chromosome is positive for the recessive sex-linked gene, that male who inherited that chromosome is always going to express that trait because there's no other X chromosome to have dominance and to essentially quell that trait that was passed on. Females would have to have a typical recessive presentation, both homozygous recessive, to actually express an X-linked recessive trait. So females, therefore, could be homozygous dominant, homozygous recessive, or heterozygous carriers. Males will either be dominant or recessive when it comes to sex-linked genes. So again, you can pass it from mothers to daughters, but females require two alleles to express. So when we start talking about inheritance patterns for males versus females, you'll see the inheritance will look different depending on the offspring. So this is showing an example here for color blindness. So this is a female who is a carrier. She's heterozygous for red-green color blindness. So you can see this is her dominant allele here, her recessive allele here on the X chromosome. The male has normal color vision here. So he has normal color vision here and then a normal Y chromosome. So if you look at the offspring, we're going to see two female offspring, two male offspring as options here. So of your female offspring, you have a female that's not a carrier for color blindness, and you have a female who is a carrier for color blindness, but neither are colorblind themselves. Then your males, you're going to have a male has normal color vision or a male who has actual color blindness because they inherited essentially that recessive X chromosome. So if the question was based off, if you had a parent who had this example, so a female who carried the trait for color blindness and a male who had normal vision, and they were going to have a boy, what's the chance or what's the percentage of that boy having color blindness? 50%, right? Now, if we're looking at this Punnett square, that looks like 25% because I didn't tell you what the gender was going to be. So if I asked what was the percentage that an offspring, and I didn't specify gender, was going to have color blindness, your answer would be 25%. And so they will do that. I've seen them do that on the exam before. They will specify gender for these traits. And so then you have to think about, okay, I know I have males, so I'm only dealing with this portion of the Punnett square. I can forget about the probability of male versus female, that 50-50 chance. I can just look at my two male options. They're equal in probability that one will have color blindness, one will not. So that means I'm going to say 50%. Okay. The same would go if I asked a question saying, what would the percentage be of females that be carriers for that trait? Then you're just leading with females and you're looking, okay, it's a 50-50 split, so 50%. Typically, when they ask about females, honestly, most of the time they ask about males with these sex-linked questions, or sometimes they'll just leave off the gender and kind of make you go through that mental gymnastics of what is the total chance that this could take place. Now, where this could change is, let's say that our male actually started with said trait. Do you have a question or just kind of, okay, I just wanted to make sure. You're good. So let's say let's say he was colorblind, and let's say our female still was a carrier. So then we started with this trait here. Then we're still going to see our female here. She'll just be a carrier. We will then see a female that has color blindness. We will see then we have a male that's not colorblind, and we'd still have a male that's colorblind. So if you had a male that was positive for whatever disease that was sex-linked, that reproduced with a female who was a carrier, that's when you can see phenotypic expression in females, because then you're going to get the X chromosome from your male if you're female, and then there's a chance, an opportunity that you'll get the one that has the actual condition on it, 50-50 shot from your maternal component. Okay. But the male would actually have to have the condition for this to take place. Questions about sex-linked inheritance patterns. I encourage you to just do, even though it feels very redundant, do a few practice like Punnett squares on your own at home and put it to a disease. So choose an autosomal dominant, choose an autosomal recessive, choose a sex-linked disease. I'm going to give you some lists here in a moment, and practice like, okay, if we have this pattern of inheritance or this pattern of an expression, what would it look like for the offspring? Because it will help it sink in your brain. So then on the test, you're not like, Punnett square. Like, oh yeah, okay, this is the condition, this is the situation. I can apply this to any type of autosomal dominant or any type of autosomal recessive or any type of sex-linked. And it's just like almost, you don't have to think about it. It's almost automatic. I see a question here. Is multiple allele inheritance the same as codominance? It's slightly different. Codominance, that's a great question. Codominance, you have two separate antigens or two separate phenotypic expressions. They're both being expressed equally, versus multiple allele inheritance is incomplete dominance, where you will have your homozygous dominant will express one phenotypic trait, your heterozygous will express a different phenotypic trait, and your homozygous recessive will express its own phenotypic trait. So they won't be expressing the alleles or the genes equally. They will just be having three separate phenotypic expressions. I hope that helps kind of separate the two. Let me know online if it doesn't. Okay. All right. Any questions about inheritance before I talk about some effects from the outside world? Okay, awesome. All right. My last time I taught genetics, we had to do a bunch of like math. So this is much better. I like that. All right. So there's lots of things environmentally that can affect normal gene expression or development. And so we're going to look at a few things. Maybe. Yeah. So when it comes to regulation of gene expression, I do want to look at a few different components. There's 3 levels of control found in the human genome. The first level is our protein coding genes. The second level is, or second layer is our small non-coding RNA segments, which is SNC RNA. And the third layer is epigenetics. And we think about epigenetics probably the most. We talk about that methylation, acetylation. We've all heard those traits or heard those things before. So we'll talk a little bit about them today. The protein coding genes recognize that of the cell's DNA, right? All of our cells have all of our DNA in the nucleus, right? It's just each cell is only expressing a certain component of our genome at any given time. But of that genome, even of the genome that can be expressed, only 2% of a cell's DNA is actually like expressible protein coding genes. 98% is just other stuff that's not coding for proteins. DNA is your full blueprint for protein synthesis, but has a lot of stuff that's just kind of extra, almost busy work, or we haven't figured out what it encodes for yet. That's the other component. So the second layer here, this is the small non-coding RNAs. This is the non-protein coding DNA. This is helping with control of what we are going to encode and how we encode it. It's about 80% of the genome. And then epigenetic marks, these are the proteins, histones, chemical groups like methyl groups and acetyl groups that actually bind to DNA. And they'll affect the way chromatin's packaged, and they'll also affect how we express certain genes or don't express certain genes. So first, looking at our small non-coding RNAs. So I kind of skipped the protein coding genes because that's what we've been talking about. And now we're talking about the small non-coding RNA regions. So these microRNAs are an example, or small interfering RNAs, PW interacting RNA. There's a bunch of them. They all have their special RNA names. MicroRNA is probably the largest category or the most common expression you'll see as an acronym or a abbreviation for small non-coding regions. Their function is to decrease expression of a particular gene, and they will essentially join forces with enzymes and target gene regions that might need to be broken up into smaller pieces, so they can actually destroy target mRNA. They can inhibit translation of target mRNA, or they can silence a gene that encodes or that targets mRNA. Why would we care about having these microRNA regions? Why would we need to silence genes? So they're not turned on all the time, right? Because what happens if genes are turned on inappropriately? Pathology, right? Yeah, because anytime we have cell division or replication or translation transcription that's happening at a constant high, fast rate, the chance for mutation and damage or disease increases, right? Anytime you repeat something over and over, if you repeat something 500 times, your chance that you're going to miss a step is probably pretty highly likely, especially if you're doing something like really fast, like DNA does, like replication. Small non-coding RNAs, part of what they do is they inactivate these transponons, which are genes that replicate themselves and can insert themselves into distant DNA sites. These can essentially disable or hyperactivate genes. So they can turn them off or they can turn them on and make them hyperactive. In development, this is really important when we're trying to take a multipotent stem cell and decide that we're going to create now an arm, and then we're going to create a heart, and then we're going to create skin. We have to decide to turn on or turn off certain areas of gene expression in that developmental portion. And so small non-coding RNA regions help with that developmental process. It's how we're able to go from our big stem cell down to all the different cell types of the body. They also can help fight off certain infections, specifically viral infections. They could be used as treatments or tools for treatment of disease. So we can target these small non-coding regions, help support them or silence them so that certain genes are more or less active. And mutations we can see in cancer and even in schizophrenia, as long as a bunch of different disorders. Really, what I want you to know about these ones, the small non-coding RNAs or microRNAs, that they're going to help with regulation of genes. They're going to either turn up or turn down the expression of certain genes. And really, we see them used normally during development. We see them used against pathologies, but normally you'll see them used during development. Epigenetic markings, I already mentioned, but this is our third layer of control of gene expression. The first layer was actually just not many proteins are encoded for in the genome. The second was these microRNAs. The third is epigenetic markings. I already mentioned methyl and acetyl groups are really the chemical tags that are used, and they're going to tell the body or tell the cell whether that specific part of DNA is available for transcription, that'd be acetylation, putting on an acetyl group, or silenced, methylation. And so this is how each cell knows what portion of the genome to replicate at any given time. If a cell doesn't have an epigenetic mark, or if it's placed at the inappropriate place, it could set the cell up for cancerous changes or even devastating illnesses that are non-compatible with life. We know that epigenetic changes can happen, they can start happening in utero, and in fact, they can even start happening in utero of the grandparent of the eventual mother of the eventual fetus. So this is where we can see kind of this ancestral health pattern being passed down, because we can actually inherit our epigenetic markers from our parents, our maternal and paternal chromosomes. So we can get some of that epigenetic markage when we are essentially a developing fetus. At some point in development, ideally, we wipe out all the markers and we start fresh. And that's so that we can have that multipotent stem cell at the top. And then we start putting epigenetic markers back on to silence certain genes. But sometimes that process of wiping the slate clean doesn't happen fully appropriately. And so then we can inherit certain things that are silenced or certain things that are turned on inappropriately, activated inappropriately. This is kind of the genomic imprinting. So certain genes tagged with methyl groups during gametogenesis can identify them as maternal or paternal, normal for development, and it allows it to express only a mother or father's gene. And each generation embraces imprinting ideally when you produce new gametes. But again, not always. Pathologically, you may not. I think this is just, this is an example essentially. So just giving you some examples of what that could look like. So deletions in chromosome 15, disorders with two different symptoms. Prader-Willi syndrome is seen if a mutated gene inherited from the father is expressed, versus Engelmann syndrome is seen if it's from the mother, the maternal. So it can even present differently clinically. You also can inherit DNA and genomes and things like that outside of the nucleus. Mitochondrial DNA, mitochondrial genomes has its own genome. There's 37 of their own genes in mitochondria, and we call that mitochondrial DNA or mtDNA. It's transmitted to embryo by mother in the cytoplasm of the egg. So our mitochondrial DNA is our maternal mitochondrial DNA. So we do not inherit paternal mitochondrial DNA. Errors in mitochondrial DNA are rare disorders. These would be your zebras. You can see issues with cellular respiration and oxidative phosphorylation. You could also see issues with maybe Alzheimer's or Parkinson's disease, so inheriting some things that could increase your risk, leading to muscle, neuromuscular, or neurologic problems later in life. So that's really the kind of two areas you could see problems with mitochondrial DNA, but they are more rare in regards to these inheritance patterns. Okay. That was our Gen Bio review of the genes. And now I want to go into some path. Yeah, go ahead.
There is a something saying that some X chromosomes in genetically females, like one of them is active and the other one's inactive.
Correct.
How does that? I mean, I know it depends on the cell line, but are we going to be asked about like the active, inactive, like?
You won't be asked that level of depth. You won't be asked that level of depth of genomics or any genetics component. You are correct. There is one maternal X chromosome that is active per offspring and one that's inactive. And so there's one that specifically our X chromosome that we're receiving our input for all those things that encode on that chromosome itself. And the other one is silenced, and it's typically silenced via methylation. So that's probably where the connection started to happen. So we talk about that epigenetically. They're not going to ask the differentiation between the two. Yeah, it's a good question though. Yeah,
No, it's not.
totally. Their main questions are going to come from asking you about recessive, dominance, autosomal, or sex-linked. Those would be high yield things to learn now. If you want to take it a step further, if you are, when we go through these conditions, if you're able to keep these in your brain and remember that they're dominant, recessive, or sex-linked, then go down the next step and start learning the SNPs, the specific genes, the little acronyms. But I would start with this first. I wouldn't learn the acronyms before you learn this list, because this will be more high yield. So this is your autosomal dominant mnemonic, very powerful dominant in humans. So you have von Willebrand disease. I wouldn't worry about von Hippel-Leydig. Pseudo hypoparathyroidism, dystrophic myotonica. You also could see this as myotonic dystrophia. So you could see them switch. Osteogenesis imperfecta. I wouldn't worry about this one. Osler, Weber, Rendu. Marfan's, intermittent porphyria, neurofibromatosis, achondroplasia or adult polycystic kidney disease, Noonan syndrome, tuberous sclerosis, hypercholesterolemia, know that one, Huntington's disease, know that one, Hockham, know that one, hereditary spherocytosis, hereditary non-polyposis coli, and hereditary hemorrhagic telangiectasia. So of this list, von Willebrand's, pseudohypoparathyroidism, dystrophia myotonica, osteogenesis imperfecta, Marfan's, intermittent porphyria, important to know those first ones. So you're very powerful dominant. So you might as well learn neurofibromatosis to give yourself the N, adult polycystic kidney disease to give yourself the A. I really wouldn't worry about this other one, but if you need another N, you can memorize it just for your acronym. It won't be tested. Neither will tuberous sclerosis, but if you want your T to make the acronym, that's fine. And then for your last one here, these two, Huntington's and Hockham and hypercholesterolemia, these three I would know. So not everyone on the list, but a lot of them on the list. These are your autosomal dominant disorders. It means you only need one allele, one gene, one genotype that has a dominant allele to express this condition. So if you had a parent who had this condition, you would know of it. There's not going to be a hidden transference of this condition to offspring, right? Although some things like hypercholesterolemia, that doesn't necessarily express itself, familial hypercholesterolemia, externally. So you might not know that your parent has the genetic form, right? Huntington's disease, you would know if a parent had Huntington's. Yeah.
For hypercholesterolemia, we need to know that right now types of hypercholesterolemia.
I would not put that to memory for this exam. They will not test you on that in depth. Yeah. They could test you on IDL, HDL, VDL, chylomicrons, maybe ApoB. They're not going to go as in depth of as our cardiology class did. And that's a pretty common thing. A lot of your pathologies that you've already learned in your block courses are more in depth than what you need to know in this exam. Because your block courses are technically prepping you also for NPLEX II. So keep that in mind. So not everything that you learned in block, even with a disease, you need to be able to remember for this test. Okay. Our autosomal recessive disorders, this is your A, B, C, D, E, F, G, H. So of the ones here, albinism and ataxia are important. Beta thalassemia, that's your B. Cystic fibrosis, if you can remember congenital adrenal hyperplasia, great, but I'd know cystic fibrosis for sure. Distal spinal muscular atrophy or Dublin-Johnson syndrome. I've never seen Dublin tested on, or Dubin, so I would do distal spinal. Emphysema, specifically the alpha-1 antitrypsin deficiency. Friedreich's ataxia, galactosemia or glycogen storage issues, and then hemochromatosis or a homocystinuria. I would just know hemochromatosis. So again, of this list, albinism and ataxia, beta thalassemia, cystic fibrosis, distal spinal muscle atrophy, emphysema, Friedreich's ataxia, galactosemia, hemochromatosis. These would be your A through H of your autosomal recessive disorders to know. And Jenny, they may have found that some people do inherent paternal mitochondria within the past 2 years, but NPLEX is never that updated. It's at least 2 years, if not 3 years old. In fact, I think they may have just included COVID on the NPLEX exam, maybe. So keep that in mind. When I went through, they'd just gotten research on berberine and used for diabetes and hypercholesterolemia, and we started using that more clinically, and that was not recognized on the test at all as an option. And it was listed as an answer, but we knew that since that research was so new at the time, it would not be the correct answer on the test. And in fact, it was not. So it is a couple years old. Not to freak you out more. Sorry, I was like, oh, okay, I think I worried you all. Okay. X-linked recessive, your Graham-Bell is your mnemonic for your X-linked recessive disorders. So your G6PD deficiency, retinitis pigmentosa, albinism, it's on here too. Autosomal versus not. I would lean into it being more autosomal than X-linked, but can it be sex-linked? Sure. Hemophilia, no, I take that back. I lean into it more as being sex-linked, not autosomal. through your albinism. So G6PD, retinitis pigmentosa, albinism, hemophilia, adrenoleukodystrophy. I don't know that condition. You won't be tested on it. I can tell you that much. Menkes disease will not be on your test. Blindness, color blindness could be. Ehlers-Danlos could be. Lesch-Nyhan syndrome, no, and Lowe's disease, no. So of these ones here, really G6PD, hemophilia, color blindness, I would say these, and Ehlers-Danlos, those ones could come up more commonly. Are there more? Of course, there's tons, right? These are just common ones that are tested on, or common pathologies that people want you to know. These are commonly tested on USMLE 1 as well. Okay. Now there's more. So this is, if these ones, this like mnemonics are kind of making sense in your brain, and you want to take it a step further, then I have some actual conditions, certain mutations, and if they have like a component of recessive or dominance or things like that. So cystic fibrosis, that gene is often tested on. So the CFTR gene, it's on chromosome 7. It's autosomal recessive. Duchenne's muscular dystrophy, it's your dystrophin gene, that DMD that we already saw in our test. It's X-linked recessive. Fragile X, Huntington's, Friedreich's ataxia, and myotonic dystrophy all have gene repeats. And so that's kind of interesting. They'll have these sections of 3 amino acids that repeat over and over. We call them trinucleotide repeats. I wouldn't necessarily commit to memory these genetic repeats like CGG, CAG, GAA, CTG. I've seen it tested once, and I thought it was a mean question. But I think it is fair to say that those 4 diseases are linked together, and the fact that they all do have these trinucleotide repeats as part of their genetic makeup. It's part of what the gene mutation is. You can see Fragile X syndrome, X, it's X-linked dominant. Huntington's autosomal dominant, as well as myotonic dystrophy versus Friedreich's ataxia is autosomal recessive. Your Down syndrome, your Edwards syndrome, and your Pateau syndromes, all these syndromes are all your trisomies. They all are going to have the 3 chromosomes where there should be 2. The trisomy for Down syndrome is affecting chromosome 21, Edwards 18, and Pateau 13. Of who is more common, Down syndrome is the most common, Edwards the second common, and Pateau the third common. And then both Edwards and Pateau are really incompatible with life. Typically, people who are born with them are dead before the age of 1 or by the age of 1. Versus Down syndrome, you can be obviously compatible with life with Down syndrome. So recognize these are the 3 trisomies in order of commonality. So Down's being the most common, second, third. And then the only one that really could have survivability would be Down syndrome. The other 2 would be not compatible with life typically after a year. Creux-Duchat syndrome, you won't see it really clinically commonly, but it's often tested on. It's tested on because it's a deletion of part of a chromosome, the short arm on chromosome 5. This really only is fun when you actually write things out genetically, because you get to write these like capital or small letter P's and numbers, and that's when it gets fun. But for the purposes here, you don't have to know how to do that for this test. So just know that this is a deletion of part of a chromosome 5, the short arm, not the long arm. Klinefelter syndrome is having 2 X and a Y. So you have the genome essentially for being female and male all in one. You get essentially an extra X chromosome. DeJorge syndrome, this is actually saying the deletion, so I wouldn't worry about this, but it's affecting chromosome 22. Breast cancer, BRCA, and hemochromatosis, HFE. These would be the ones I would know. Celiac disease is a good one, although I haven't seen it tested on as lately, although for a while there was like a pretty, when they first found the genetic linkage, it was tested on every single time. So that could be another good one to commit to memory. That's not on here, but I would say these are commonly tested. So if you're able to kind of differentiate and understand these conditions as a starting point now, you'll be well off for the test. Okay. That was a run through of general genetics concepts and kind of my high yield list. We're going to go more into cell stuff here in the next hour, but any questions before we break? Okay, genetics is part of biochem. Biochem typically is more of the section than genetics. So I would say if you're able to figure out your dominance, your recessives, and your sex-linked, you'll be pretty good for that section. Great. All right, we'll take a 10-minute break. So we'll start back up right at 9 o'clock, and we'll be moving into our next section, which is going to be biology of cancer, but really it's more cell path because they'll ask you about some of these cell pathological conditions, metaplasia, dysplasia, et cetera, on your test. So that'll be next.